Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observations and commentary on the JCO article, a Phase 2 and biomarker study of the dual MET-VEGFR2 inhibitor ferretinib in patients with papillary renal cell carcinoma by Tony K. Choueri et al. My name is Edwin Posadas, and I am the Medical Director of the Urologic Oncology Program at the Samuel Ocean Comprehensive Cancer Institute at Cedars-Sinai Medical Center in Los Angeles, California, USA. My oncologic specialty is medical oncology. In this issue of the Journal of Clinical Oncology, Choueri et al. Present an important study investigating the role of ferretinib in the treatment of patients with papillary renal cell carcinomas, or PRC. PRC is the most common form of non-cleared renal cell carcinoma. Given the relative rarity of the subtype of RCC, only a limited amount of information exists to guide treatment decisions. In addition to unique histologic features that distinguish PRC from clear cell RCC, there are unique molecular features associated with the disease. In particular, there is overexpression and overactivation of the MET kinase through a variety of means, including mutation and copy number aberration. While the presented study failed to demonstrate activity as defined by the primary endpoint of overall response rate, the investigators do show potent clinical activity in the small sample subset that justifies additional work in this area. In comparison to currently used agents targeting the vascular endothelial growth factor receptors or the mammalian target of rapamycin, ferretinib appears to be more clinically active. While this agent does inhibit a number of non-VEGFR, non-MET tyrosine kinases, particular emphasis in the study is placed on MET, given the body of knowledge of MET variation in PRC. The development of targeted therapeutics in cancer has not yet met the high expectations of patients and physicians. The majority of this disappointment appears to result from the rather limited rationale for the advancement of certain agents in particular disease systems, or an inappropriate understanding of the biology of the system. The clinical studies performed often fail to answer an important question. Is the relevant molecular target present in patients? And does this correlate with disease biology or response to therapy? The authors of this manuscript are to be congratulated for taking their observation beyond a generalization about PRC and aberration of MET expression or function. Rather than assuming that any variation in MET signaling is equivalent, the investigators stratified patients by the type of MET abnormality present in the pool of responders. In addition to duplication of chromosome 7 and other copy number aberrations, there are more than 60 cataloged mutations in the MET gene. A proportion of patients have these mutations in MET in the germline, whereas others may acquire MET mutation through a variety of means. This study points towards the importance of the MET aberration in the germline DNA. Interest in MET as a target of anti-cancer therapy has grown. In particular, there are several groups that have proposed that activation of MET may be an adaptive response to certain forms of anti-cancer therapy. In particular, the MET-EGFR interaction has been pursued as a means of overcoming EGFR therapy resistance. Currently, there is growing interest in the cooperative activity of MET and VEGF as cooperative signals in tumor invasion and angiogenesis. These types of cooperative signaling interaction may account for some of the biological activity seen with other multi-targeted tyrosine kinase inhibitors, such as cabozantinib, which has shown activity in castration-resistant prostate cancer, medullary thyroid cancer, hepatocellular carcinoma, and other settings. These findings bear high value if MET functions primarily as a somatic adaptive response to external pressure. The MET-VEGFR interaction in the tumor microenvironment bears particular interest given the high rate of pulmonary emboli in this study, perhaps implying a greater degree of vascular disruption. Interestingly, the investigators in this study note that germline mutation bears stronger predictive value than somatic mutation. This finding has both clinical and scientific import. First, though studies restricted to germline MET mutation may potentially strongly validate the importance of this observation, they would limit the scope of clinical benefit severely without taking advantage of the observation that non-germline mutant patients did experience benefit from therapy, though to a lesser degree. Second, the importance of germline mutation, as compared to somatic mutation, must be considered and may have importance in understanding the functions of these genetic aberrations. This may lead to important insights that may guide therapeutic development. In this study, classification of patients was restricted only to genetic information about MET, which may not fully reflect the activity of this pathway in patients, especially given findings that gene expression and protein expression levels do not strongly correlate with MET. Given the limited tools, however, for detecting the presence of MET in tissue and the lack of consensus on reliable means of defining its activation state, the genetic approach is to be considered the most feasible to employ at this time. As presented, the study introduces a number of important clinical and translational questions that will advance the field of therapeutics in the clinical entity of PRC, but also in turn will enhance the field of rational therapeutics and personalized medicine. First, while germline mutation predicted higher rates of response, half of the patients with germline mutation still did not experience benefit, and the reasons for this are poorly understood. Second, do these findings suggest that germline mutation status is more important than somatic mutation status in general? Third, do these findings imply that the majority of benefit from these forms of therapy are more dependent on the tumor microenvironment than the tumor itself? Fourth, non-germline mutations appear to still correlate with benefit at a higher rate than wild-type MET. Is there a molecular similarity in MET or ferritinib-related targets that accounts for this? In conclusion, Choueri et al. Have identified a highly active compound in a variant of kidney cancer using a highly translational investigative approach that sheds additional insight into the mechanism of benefit by identifying the subset of patients with a germline mutation in a putative target gene as a group that may experience high rates of benefit from therapy. The findings and approach from this study may be generalizable to other non-PRC models where MET may play a critical role, either in the tumor or the tumor microenvironment. Given the introduction of a number of agents capable of inhibiting MET, translational approaches such as that presented by these investigators must be taken to understand the biological underpinnings of the clinical impact of treatment. This concludes this JCO podcast. Thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.